let's quickly revise the cell cycle. There are two main phases: the growth phase, which we can also say the interphase, and the division phase, which is also called the M phase. Now, during the interphase, the DNA and the rest of the cell organelles are duplicated. Now, the process of cell division includes two sequential processes: mitosis and cytokinesis. Now, in the interphase, there are three stages: the G1 phase, the S phase, and the G2 phase. G1 phase is the longest phase. It synthesizes RNA and proteins. The S phase is when the DNA and the centrosomes are duplicated, along with other cellular organelles. Now, the G2 phase is the shortest phase, which proteins that are necessary for the cell division are synthesized. Mitosis. There are four phases: prophase, metaphase, anaphase, and telophase. Prophase. The chromatin condenses and chromosomes become visible. The centrosomes migrate to opposite poles and begin to synthesize the mitotic spindle. The nuclear membrane begins to break down and the nucleolus disappears. Metaphase. The spindles attach to the chromosomes and move them to the center of the cell. This forms the metaphase plate or the equatorial plate. Anaphase. Centromeres break and sister chromatids of the chromosomes separate. Spindle fibers pull them to opposite poles. Telophase. This begins when the chromatids reach the pole of the cell. Now, chromosomes decondenses once again into chromatids. Nucleolus becomes visible again, and the nuclear membrane is reformed. After mitosis divides, we have the cytokinesis. It is when the cytoplasm divides. In animal cells, a contractile ring forms and is made of actin and myosin. In plant cell, it occurs by the formation of a separating wall, called the phragmoplast. Mitosis ensures that the daughter cells remain the same number of chromosomes as their parents. So in short, in mitosis, for eukaryotic cells, it helps to grow and repair tissues. In prokaryotic cells, it helps for reproduction. Meiosis. It is a characteristic of organisms with sexual reproduction. It reduces the number of chromosomes into half, and it also increases the genetic diversity. There are two types of divisions: meiosis 1, meiosis 2. In between them, there's a short interphase, however, no DNA synthesis takes place. There's prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 is the most important stage. In this phase, a crossover occurs between homologous chromosomes, which helps in DNA diversity. The nuclear membrane disappears and a mitotic spindle forms. Metaphase 1, spindle attaches to the homologous chromosomes, moves them to the center of the cell, metaphase plate. In anaphase 1, homologous chromosomes separate. In telophase 1, nuclear membrane reforms and the mitotic spindle disappears. Chromosomes partially decondense. Moving on to meiosis 2. Nuclear membrane disappears and mitotic spindle forms. In metaphase 2, mitotic spindles attach to the chromosomes and moves them to the center of the cell, forming the metaphase plate. Anaphase 2, centromeres break. Sister chromatids of the chromosomes separate. Telophase 2, chromosomes decondense again to form chromatin. Nucleolus becomes visible again, and the nuclear membrane is reassembled. In conclusion, mitosis produces two genetically identical daughter cells, whereas meiosis produces cells genetically unique from the parent and contain only half as much as DNA, which means 4 haploid daughter cells. Now that is a quick recap for today. Good night.